I'm going to more focus on the lead optimization phase, okay, and talk about a project that we did a lead optimization on that led to a compound that we have now entered into the clinic. Okay. Before I get started, I do have a disclosure, okay. The program I'm going to talk about today was partnered with a pharma company. There are milestone payments associated with the deal that go to MD Anderson. I am an employee of MD Anderson, a co-inventor of the final molecule with potential financial benefits through our revenue sharing program. Okay. So I'm representing the Therapeutics Discovery Division at MD Anderson. So you can think of us as an academic drug discovery group, but we run ourselves as if we are a pharma company. Okay. Now what that means is that we're made up of a bunch of different subgroups, each with a particular expertise in one small piece of the drug discovery process, and we all work together to discover and develop drugs. That's very similar to the sort of environment you might have in a big pharma company. Okay. We're about 75 people at this point, with a lot of us having worked in industry before we came to MD Anderson. Okay. The project I'm going to talk about is our glutaminase program. Okay. Glutaminase is an enzyme that converts glutamine to glutamate. Okay. That glutamate then gets converted to a bunch of other molecules that are important for cellular processes. Some tumors heavily rely on this glutamine-derived glutamate to support their survival. There's a fair amount of literature out there that discusses that glutaminase inhibition will induce antiproliferative effects both in vitro and in vivo. Okay. There's also one company, now two people that are entering the clinic who have a compound that they've done some clinical testing on that showed some anti-tumor effects. Now, the reason why we were excited about glutaminase as a target for inhibition comes from some studies we did that showed a differential effect on cell growth between different cancer cell lines when treated with glutaminase inhibitor. Okay. We get excited by targets like this that basically it matters what your specific tumor type is, whether or not you have activity against with an inhibitor, because that's something that we hope to figure out why you've got that differential activity, and does that translate to a different patient population that would respond to a drug versus one that wouldn't. Okay, this gets into the idea of personalized medicine, and that when we treat patients, we want to treat patients that we know will respond to a drug. And so if we know at a very molecular level what sort of patients will respond to a drug, that makes an exciting target for us. Okay. What I'm showing here is basically profiling glutaminase inhibitor across a variety of cell lines, okay, where you see a very clear no responder in some cell lines and a strong response in other cell lines. An important point here is that in every single one of these cell lines, the glutaminase inhibitor testing is doing the exact same thing, okay? It is stopping the conversion of glutamine to glutamate. Okay? That's what I'm showing here in this bottom left box here. These are two different cell lines, one that is a responder, one that's a non-responder. Okay? These bars represent a ratio of glutamate to glutamine. In the absence of inhibiting this process, the relative amount of glutamate goes up, okay, and so your ratio of glutamate to glutamine is high. Okay. When you treat with an inhibitor, you block this reaction, there's less glutamate relative to glutamine, you expect that ratio to go down. Okay. When you look here, regardless of whether you're in a responding cell line, which is this A549, or non-responding cell line, which is this A727, you're doing the exact same thing at a reaction level. Okay. However, you have very different responses. When you look sort of at the biochemical level of what's happening, what the difference is between these responders, or most of these responders and most of these non-responders, what we find in this case, you have mutations in one or the other of these proteins, KEAP or NRF2. Okay? I'm not going to get into biology, okay? These are proteins that are involved in the regulation of glutathione synthesis. Okay? What that's important for from the big picture perspective is that is a common mutation that is seen in tumors. These are non-small cell lung cancer tumors we're talking about. And it serves as a patient identifier for the sorts of patients that would benefit from the glutaminase inhibitor. So on the biology end, that was very exciting, and they wanted to then create a glutaminase inhibitor program to make drugs to take into the clinic. Okay, so in our sort of pre-questions, there are a number of questions just had to do sort of big picture about lead optimization. I'm going to intersperse this talk with a couple sort of big picture slides of what it is we're looking for when you're doing a lead optimization program. This is the first of those slides. This, as medicinal chemists, is what we're looking for. Obviously, you need to be a potent inhibitor of your primary target. Okay? And equally, obviously, you then want that to have good cellular activity. What sometimes gets lost in this is inhibiting the primary target and getting good cellular activity aren't always necessarily a cause and effect relationship. So you also need to have some assay that tells you that you are actually doing the same thing in cells that you did in your enzyme assay, and that correlates to your cellular activity. Okay. You want a high selectivity over off targets. This is both in terms of understanding what's going on, but also in terms of safety. Okay. The more things you're hitting, the more likelihood that you're going to do something you don't want to do. There are plenty of programs that go after polypharmacology. This is not one of them. Okay. You want physical, chemical, and pharmacokinetic properties suitable for human dosing. Okay. Again, in terms of what you consider suitable for human dosing, that will vary in terms of from outfit to outfit, but in our case, we're looking for oral dosing where we're developing pills. And then obviously you want to have efficacy, and you want efficacy that is happening because of the mechanism of action that you're proposing. And then we want a favorable safety profile. Okay. A very important thing in any lead optimization process is to have good assays that really confirm that your molecules are doing what you say they're doing. This usually requires multiple assays. The programs you're on will all have different assays depending on what you're looking for. So the assays we use for this program are very different than what Andy uses as degraders. Okay. In this case, I won't go into all the details of the assays, but there's a simple enzyme assay that we're looking at for inhibiting this glutamine to glutamate conversion. Okay. We have a similar cell assay that basically measures the same thing, and that we want to correlate to a cell phenotypic assay, which is looking at inhibition of proliferation of the various cell lines. Now, the assays are very important, but the other obviously important thing is you need to have a chemical starting point. Okay? You can't optimize a lead if you don't have a lead. Okay? For this particular program, our lead came from the literature. Okay? There was a molecule published about 20 years ago by a company called Elon called Beptis that inhibits glutaminase. Okay? This is far from a perfect molecule. It actually has quite low solubility, and I'll explain in a few slides why that's so important. It also has fairly low metabolic stability. Okay. It did have two things we really liked about it as a starting point, though. Okay. The first bit is that it's a very selective glutaminase inhibitor. It's also selective for one particularized form of glutaminase, GLS1, which is the one we're trying to hit. Okay. It doesn't hit a separate isoform called GLS2, and it doesn't, to the best of our knowledge, really hit anything else that we've seen molecules this type profiled against. The second really important thing about this is that there was an available crystal structure. Okay. We're going to be designing analogs of our own that have hopefully improved properties. That is greatly helped if we have a crystal structure that's a snapshot of how the molecule binds. Okay. And that's what I'm showing here. Okay, so Beptis is an allosteric inhibitor, meaning that it's binding to a separate site than what glutamine binds to. It's a very cool structure. To begin with, it's kind of this oligomer of glutaminase molecules. Okay, there are 4 glutaminase molecules that are binding to 2 inhibitor molecules. Now, I'm going to, if I can figure out this. Okay, so the inhibitors are binding at the interface of 2 glutaminase molecules. The main thing we're paying attention to here for our design is this hydrogen bonding network shown in the yellow dotted lines. Okay, that's these things right here. Okay, we think those hydrogen bonds are very important for getting activity. So when we went into our design of our own molecules, that was a big focus of the early lead generation activities. Okay. So our strategy here, again, focusing on this hydrogen bonding pattern. Okay, we're looking at different heterocycles that would maintain that hydrogen bonding pattern. Okay, a second part had to do this part in red here, this linker. Okay, this linker is one of the parts that gives metabolic instability to these molecules. Sulfurs are very easily oxidized up to sulfoxides and sulfones. Okay, so we wanted to modify that. And then what we consider our end game is fine tuning of these wings in purple here. Okay, what you can do in this area in terms of potencies, you've got a lot of flexibility of what you can put in here. So really, these were more about helping with properties of the molecules than anything else. So we're not actually in the lead optimization phase yet. Right now, we're just trying to look for leads identified. We looked at a whole bunch of different scaffolds, greater than 30. Okay. At the very early stage, we just were looking for potency. We took 3 front runners of promising potencies for some further optimization of the leads. And these 3 all have multiple examples with enzyme IC50s less than 25 nanomolar. Okay. Now, I've already mentioned a couple times that it's not enough just to get something that's active. Okay. it should be active for the reasons that you think it is. It helps you with your later design principles if you actually know what's going on, okay? So an important thing here is we have activity. Are we confirming that we're getting that activity because we're binding to that site that I already showed? Okay? Because if it's not binding to that site, it's going to be very hard to design things based on that original crystal structure snapshot. Okay? Now, the easiest way to confirm that binding is to get a new crystal structure, and we were able to do that for one of the series, which I show here. Okay? A challenge with this program is that it's actually fairly hard to get crystal structures of glutaminase with inhibitors bound to it. Okay? So, and this is a whole separate group that did this, but I think it's a neat trick. For the molecules we couldn't get, or the series that we couldn't get crystal structures for, we actually used this trick of designing cell lines that would have mutant forms of glutaminase enzyme. Okay? So, turns out you can mutate one of the residues in that binding pocket that we're targeting, and the glutaminase itself will still have all the same glutaminase activity, but it will no longer bind to inhibitors at that particular binding site. So, if we can't get a crystal structure, we run things in a wild type line and a mutant line, and if it has activity in the wild type and then we abolish that in the mutant, then we take that as evidence that where it binds is where that mutated residue is. Okay, another big picture slide. We're going to talk a little bit about then optimizing those three series and focusing down on one of them. Okay, so just for people who are not familiar with optimization, at its very most general level, it just means installing all of your desired properties in that single compound you think can be a drug. Okay, there are all sorts of things that fall in the bucket of desired properties. Okay, most of them fall into three bins, which is obviously their biological activities, and that would include off-target activities, so safety. Physical chemical properties, things like solubility and permeability, and pharmacokinetics, okay, which would include, but is not limited to, general stability to metabolizing enzymes in the body. What me and my group do is we design and synthesize molecules that we then give to all those other different subgroups I talked about, and they run assays that tell us the information in these categories. Okay. Sometimes we'll get, one of these really right, but the rest of them will be suboptimal. So the data will come back to us. We'll design new molecules. We'll send them back to all of our biologists and pharmacokinetic scientists, and they will give us the next data round to tell us if we improved on the things we were trying to improve. This is a highly iterative process. Sometimes you fix one thing and something you thought you had nailed, all of a sudden it is no good anymore. For this particular program, this overall ended up being probably about a 2-year process and made well over 1,000 molecules in this. Okay. So I mentioned there were 3 series we were initially interested in. It's super important to have multiple series, okay, because many of them will drop out, okay. In this case, what I'm showing here is just simply a simple graphic of a year's worth of work on this program, mapped up against cellular potency. It's just one parameter to talk about. One thing should be very clear is that this first series we're working on in that year versus the other 2, we could not get things to these same potency levels, okay? And that turned into a no-go for this series. Okay. This middle guy, okay, we worked on quite extensively. It had great cell potencies, but actually it had very poor stabilities when you dosed this in animals. Every compound in here, even if all your in vitro data said it would look good, tended to just not show up in the bloodstream when you dosed the animals. Okay. Ultimately, that led to a no-go for this series because we had this 5,5, which had the excellent cell potencies and had moderate stabilities, and we thought this was going to be worthwhile and we'd be able to turn this into a drug. Okay, from a big picture perspective, this is the most important slide. Okay, the take-home message is always potency is not enough. You also have to have a drug that is able to reach its site of action. Okay, in our case, we're looking at an oral drug, which means what happens after you take a pill? How does that drug that's in that pill get to, in this case, probably a tumor somewhere in your body? Okay? So the number one thing, it needs to have solubility. Okay, it's going to be sitting in aqueous media, in your stomach, in your intestine. Okay, if the molecules don't dissolve to their sort of individual components, they're not going to be able to get anywhere. They're just going to pass through your system. Once they dissolve, there's a variety of bilipid membranes it's going to have to get through before it finally gets in your bloodstream. Okay, so it has to be permeable. It has to be stable to all the things your body knows how to do to get rid of that drug. Okay, the 2 primary ones being straight out metabolism, destruction of the molecule, typically in the liver, or excretion, which is typically through the kidney. Okay. Once it gets through that, then it can distribute around the body and hopefully get to where you want it to go, which in this case will be wherever your tumor is. Okay. The take-home points here, Drug must be soluble and permeable with some stability against elimination. As we go through this program, you'll see that I'm not going to talk much about permeability. Okay, permeability was typically not a problem for us, but solubility and stability to some extent, those were the big things that we had to deal with. Okay. So we played around a bit with this 5,5 series and came up with this molecule that we sort of called our lead molecule for a while. Okay. This molecule has really good enzyme potency and cell potency. Okay. I'm not going to go into the details of what these clearance number means, but basically we had moderate in vivo metabolic stability. Okay, if you injected it into a rat, directly in his bloodstream with an IV dose, compound would stick around. Okay, big problem though is that if instead you fed this compound orally to a rat, okay, absolutely none of it would show up in the bloodstream. Okay, it had what we refer to as a 0% bioavailability. Okay. So the issue here is that this is a poor solubility compound. Okay, so 7 micromolar in this particular viral media is a low number. Okay, so I talked about the very first important thing was your drug has to dissolve after you take that pill. Okay, this thing is not dissolving; it's just passing through. Okay, so that was a big thing we had to deal with in our lead optimization efforts, really improving the solubility. And while we had moderate in vivo stability, that's fine when it's your first lead molecule. You definitely still have to improve upon that to get an actual drug. Okay, so I'm not going to show lots of structures and data in this talk. It's more of a high-level talk. This is a summary of the sort of changes we looked at to improve those properties. Okay. I've got 2 aromatic, what I'm calling wings. Okay, aromatics were always the best for potency. Okay, but it turns out you can improve your properties quite significantly by just chopping the whole thing off. You lose some potency, but you get better solubility. Okay, and ultimately that ended up being a smarter way to go than just being married to the potencies. You couldn't get rid of both the aromatics, then you just wouldn't have hardly any potency at all. But we did find you could do further optimization for properties with these guys. Okay. There's a fluorine here. Fluorine is fairly well known in medicinal chemistry as something you can put onto alkyl pieces to improve their metabolic stability. Okay. In this particular case, this came from some structure-based drug design, looking at that original crystal structure and sort of modeling our compounds into it. And it wasn't complicated. It just showed that there was clearly space to put a fluorine, on this linker. Okay, and that does improve metabolic stability, also improves potencies. Okay, and then finally, we continue to look at replacements for these hydrogen bonding cores, and ultimately we found that actually replacing one of these guys gave us some further help in our properties. So the 2 I'm going to focus on in terms of our sort of breakthrough moments on this program was hacking off this aromatic ring and doing a final swap here for a very close related heterocycle. Okay, so cutting off the aromatic ring, okay, that gives us this compound here from the original one. You see we do have a loss in potency, okay, fairly significant, but we also get about a 5-fold or more increase in solubility. Okay, it's actually a bit more stable compound in terms of what's called the total clearance. It looked about the same, but what was exciting was that we had some bioavailability. Now, 3% is really, really low, okay? It's almost comical looking at it now and saying that was a breakthrough moment for us, okay? But what you're not seeing were the huge amount of compounds we made before that guy that had nothing at all. Okay, so having anything at all at that point in the program, I mean, we were hitting the ceiling. We were incredibly excited, okay? But where we really saw the boost in the bioavailability, which is really mostly to do with making a more stable compound, was when we pulled a small switch from this 5-membered triazole here to the 6-member pyridine, okay, along with a couple amide transpositions. Where now we actually get back most of our potency, similar, maybe slightly better solubility, but more importantly, lower is better here. So this became a more stable compound when you dose it to animals, and that all resulted in having a bunch more compound that actually ends up in the bloodstream when you dose orally. Okay. Now, this is actually the last structure I'm going to show you. Okay, the final candidate is very close to this. Basically, we vary this to fine-tune things. We are disclosing the structure at the American Chemical Society meeting next month, okay, but right now I'm not allowed to tell you what the structure is. Okay. What I do want to do is show you some of the check boxes for what I said we wanted to look for in a drug. So it's a potent inhibitor of primary target. Okay. If you're remembering the numbers, this is about 6-fold less potent. In cells, it's about 5 to 6-fold less potent than what we had for that original lead compound. This is kind of an important point. The best compound is very, very often not the most potent compound. Okay, it's not always just about the potency. In this case, this is less potent, but our overall properties, like, made it way better than those original lead compounds. We have good cellular activity. This is looking at inhibition cell proliferation. This is our target engagement, showing that we're actually engaging target, and we get a nice, very similar numbers across the board for these, giving us very high confidence that it's working for the reasons that we say it's working. Okay. It's high selectivity over against pretty much any off targets we ever looked at. I'm just going to show the GLS2 IC50 to make the point here. Really excellent PK across species. Humongous oral exposures. I'll show one example on a later slide, but it was the same across all species that we tested. I will show data in terms of going in vivo with this molecule in the next few slides, and obviously we brought this into the clinic, so we thought it had a favorable safety profile. Okay. So I'm just going to show some of the in vivo data for this compound. Yeah. Yes.
Studies, that's the first time you directly assess whether or not you have unexpected off-target levels in the drug or
Yeah. So basically, in this bucket here, there's a lot of different things. Okay, so there's a urifins panel. If people aren't familiar with that, is primarily looking at ion channels, GPCRs that you might hit. There's looking at a kinase panel. You hit kinases. It's looking at a variety of cytochrome P450s, both for inhibiting them or inducing them, okay, as well as other ones that we're not discussing here. Yeah. Right. So I don't know if there's a huge difference between these numbers, okay? Although we do have cases where with the really potent compounds where the cell is actually better than the enzyme, okay, which we think has to do with conditions of the assay and stuff like that. When you're talking about the tenfold, you may be in kinase world for that. It just the mode of action of this. It's an allosteric inhibitor. It's not really competing against any endogenous substrates. There's really no reason to expect it to have a shift going into cells. Oftentimes when you get that shift, it's either because of some sort of properties, like it's not totally permeable, or it's because it's competing against some endogenous substrate. And because of that, at that competition, it's not quite as potent as it is when you don't have the endogenous substrate. Okay, so you have to get your molecule to the site of action. To get to the site of action, you need to have it get into the body and bloodstream in the first place. Okay, so this is just taking this molecule at this particular dose. Okay, dosing it to mice and looking at what its plasma concentrations are. 100 micromolar concentrations is a very respectable number for just about any program I've been familiar with. To give some degree of calibration here, this dotted line here is just our cellular EC50 for this particular cell line that is a responder. This is a log scale, to be fairly obvious that we can attain exposures that are just whoppingly over where we think we need to be. Okay? This is now the same cell line, but being used to make a mouse model, xenograft mouse model, and showing that we are, when you treat with compound, we are raising up glutamine, we are dropping glutamate. That's exactly what you expect to see for a glutaminase inhibitor. I'm not showing dose response here, but this is maxing out the effect, both with our compound and with other glutaminase inhibitors. Okay. Glutamate is a feedstock for making glutathione. Okay, I mentioned before that one of our patient responder IDs has to do with mutations of proteins that are involved in the regulation of glutathione synthesis. So we definitely want to also see that we're dropping glutathione. Okay, and we do see that here. So white dots is vehicle, black is treated with compound. Low down this end is glutamate. Low down this end is glutathione, and this is all going in the reduction end. Yes. Yeah. I'll show one in the next. Okay. This is just the PD side. Okay. It's actually an interesting aside in terms of what's the importance of this versus what's the importance of how you look in a mouse model, okay, where we tend to put a lot more importance on are we getting the biology that we see, because the actual predictability of the animal models because it shrinks the tumor here, that's what's going to happen in humans, it tends to be fairly poor, okay. But that being said, everyone likes to see a tumor model, and so it is something that is done in the studies. So, interesting thing here, and this is not just our work, this is the entire glutaminase field. What's become very apparent is that in terms of shrinking tumors, glutaminase inhibitors as monotherapy tends to not do a whole lot. okay? You do get significant slowdowns in tumor growth, okay? But really where the excitement is in the field of contaminase inhibitors is what happens when you combine them with other agents. Okay? So, not releasing yet what this agent is, okay? It's another drug that's in clinical trials right now, okay? But when you combine our drug with this drug, okay, you go down from, you know, two drugs that are doing partial regressions to, partial slowing of tumor growth to ones that are at stasis or maybe regression, okay? Again, I can't give details on it. This is stuff that will be presented at a cancer conference in April. But the general idea here is that when you treat with contaminase inhibitor, there's another biochemical network that gets upregulated, okay, as a compensating mechanism. And when you treat with clinical agent X, it hits that compensating mechanism and vice versa. Okay, it was a nice piece of biology. It was done by biology colleagues to really show that this should be a rational combination, and it works quite well. Okay. I'm wrapping up here, okay, but this molecule we did partner up, as I mentioned, with a company, French company named Ipsen, okay. We partnered up with them a little under 2 years ago, and we entered the clinic a year ago, okay. I can't give clinical details. I will say we're pretty happy with how our phase 1 is going at the moment, okay. But that's about all the information I can give you guys at the moment. I apologize for that. You know, that's my teaser. I will just wrap with special thanks to the entire team, but especially Phil Jones, who's my boss and the head of discovery, okay? Kong and Gong were the ones who came up with the final molecule, and especially my co-leader in biology, Jeff Kovac.